Our protocol expands the use of G. mellonella from a systemic infection model to a model that allows bacterial administration via the natural oral route.
G. mellonella is a suitable replacement model for rodents in analyzing different hallmarks of innate microbe-host interactions, as the larvae can be used for human pathogenic bacteria.
Begin by transferring the eggs laid by adult moths to 2-liter boxes containing wax moth substrate at 30 degrees Celsius in the dark. After 1 to 2 weeks, transfer 25 grams of larva-containing substrate into fresh substrate when small and tiny larvae become visible. Synchronize the larvae after 2 weeks according to their size into groups of 30 to 40 larvae in new 2-liter containers on wax moth substrate. After 2 weeks, select larvae for the experiment by weight, using only pale, fast-moving larvae with a mass of 180 to 200 milligrams. To force-feed the larvae, load an insulin syringe with a blunt-ended needle for oral application with 1 × 10⁷ bacteria per 10 microliters of DPBS per larva. Load the syringe into a microsyringe pump. Wait for a larva to open its mouth parts before inserting the syringe carefully between the mandibles and delivering the bacteria. When all of the larvae have been fed, incubate the animals in the dark at 37 degrees Celsius between 1 to 24 hours. For larva processing, clean a safety hood.